Presenter is John Kelly, Professor of Biology at Loyola University, Chicago.
Great, thanks very much. So I'm going to be talking to you today about some work that we've been doing exploring the potential interactions between microplastics, pharmaceuticals, and microorganisms. So these are all things that are found in urban freshwater ecosystems. And so I want to sort of explain first how I think the pieces of this puzzle fit together and why I think it's an interesting topic to study. How am I advancing? Page down. Okay. So microplastics, of course, are plastic particles that are less than 5 millimeters in size. They've been documented in aquatic habitats throughout the globe, including both marine and freshwater ecosystems. When we look in urban rivers, like this is some data that we collected from the Chicago River, it's been seen by other studies as well. Fibers are one of the most common microplastic types we find in these habitats. This data is from the Chicago River. The graph to the left is surface water. The graph to the right is sediment from the benthos of the river. And you can see in both of these habitats, and the samples were taken upstream and downstream of a treatment plant, wastewater treatment plant, that microplastic fibers are one of the most common microplastic types we find. These fibers, previous speakers have mentioned, can come from the laundering of synthetic textiles. So when we launder these synthetic textiles, they release microplastic fibers, which means they get released to domestic wastewater. When the wastewater goes to a treatment plant, most of the microplastics are retained in the treatment plant and end up in the biosolids, but it's not complete removal. So there's still significant amounts of microplastic that come out of the treatment plant in the effluent and can get released to surface waters. So this is some more data we collected from the Chicago River showing that the levels of microplastic in surface water are significantly higher downstream of a municipal wastewater treatment plant as compared to upstream. And that holds for microplastics in general, as well as all these different types that I've shown here: fragments, fibers, pellets, and foam. When we look at these microplastics that we find in these urban rivers, they're colonized by dense biofilms of microorganisms. So you find microorganisms colonizing all of these different microplastic types. And when we analyze the microbiomes on these microplastics, the types of microbes that you find in the plastic are distinct from the surrounding habitats. So the microplastic microbiomes are different in taxonomic composition from the microbiomes we see in the surrounding water, as well as floating organic material. So that's my explanation of the interaction between microbes and microplastics. The next thing to talk about is the interactions between microbes and pharmaceuticals. So pharmaceuticals and personal care products are also present in domestic wastewater. Our normal use of pharmaceuticals and personal care products, whether it's surface use or whether it's ingestion, is going to result in the introduction of these compounds to domestic wastewater. And again, as I mentioned, for the microplastics, this wastewater will be treated at a wastewater treatment plant. Some of these treatment plants remove some of these pharmaceuticals and personal care products relatively well, some not so well, but in most cases, the removal is not complete. And so wastewater treatment plants can also be point sources of PPCPs to the environment. So this is some data we collected from some streams around Chicago. There's a lot of numbers up here, but the point is that this treatment plant that we studied in the suburbs was a significant point source of a number of different pharmaceuticals to the river as well. And we know from work by me and my collaborators, as well as lots of other people, that when PPCPs enter into aquatic habitats, they can interact with and affect the microbial communities that live there. So this is some data that we did where we did a controlled experiment where we exposed stream biofilms to a number of different pharmaceuticals. And we saw that these pharmaceuticals can have a significant effect on the activity of the microbial biofilms, including decreases in respiration rates, which is shown in the graph on the left. We saw significant reductions in respiration rates driven by a number of compounds, including caffeine and diphenhydramine, which is an antihistamine. And in addition to reducing respiration rates, we also saw that these pharmaceuticals could change the composition of the biofilm microbial communities. In addition, if we start talking about antimicrobial compounds, exposure to antimicrobial compounds in surface waters can lead to selection for resistant organisms. So these are some data that we collected looking at the effects of an antimicrobial compound called triclosan And we saw that when we measured triclosan concentrations in rivers around Chicago, that's the data on the left, we saw a significant positive correlation between sediment triclosan concentrations and the levels of triclosan resistance in the bacterial communities. And we modeled the same result in a laboratory experiment where we controlled and added triclosan to artificial stream communities. And we saw that exposure to low levels of triclosan could select for resistant bacterial communities as well. So these pharmaceuticals and antimicrobials can impact the activity and composition of microbial communities. They can also select for resistant organisms. So that's the connection between the microbes and the pharmaceuticals. The last piece of the puzzle then is the connection between the pharmaceuticals and microplastics. So I've already explained to you that both pharmaceuticals and microplastics are present in domestic wastewater. They can make their way through treatment plants and they're not completely removed. Work by a number of other groups have also shown that microplastics, because they have high surface area and because they tend to be hydrophobic, are really good at absorbing all different kinds of organic contaminants. So they've been shown to absorb things like PCBs, PAHs, pesticides, and then the study I mentioned at the bottom, pharmaceuticals and personal care products as well. So the microplastics and the pharmaceuticals are present in domestic wastewater. They're traveling together through the wastewater infrastructure. They end up in the treatment plant. There's lots of opportunities for them to interact and for these pharmaceuticals to adsorb to the surface of the microplastics. So this leads me to believe that there's the potential then for microplastics to serve basically as hotspots for the interactions between microorganisms and these pharmaceuticals, right? The pharmaceuticals can adsorb to the microplastic surfaces. The bacteria and other microbes like to colonize the microplastic surfaces. So these could be places where we have strong interactions between the microorganisms and the pharmaceuticals, right? On the surfaces of these microplastics. This could be doing things like changing the community composition, changing its activity, as well as potentially selecting for resistant organisms. It also could potentially be increasing the rates of biodegradation of these pharmaceuticals because they're going to be interacting with the microbes on these surfaces. So this is sort of like the idea that I'm trying to explore in a number of studies that we've been doing in my lab and with some collaborators. So I'm just going to highlight two really quick studies that were done in my lab working with some collaborators to explore whether this is actually happening, whether the adsorption of pharmaceuticals to microplastics could affect microbial communities. So we did a microcosm study working with Nina Oforji, who was a local high school student on the left, and then a mesocosm study working with Erin Adams, an undergraduate student highlighted there on the right. So in the microcosm study that Nina worked on, we took microplastic fibers that we just prepared in the lab of 3 different polymer types, acrylic, nylon, and polyester, and exposed them overnight to this antimicrobial compound called triclosan. And what we saw was the different fiber types adsorbed triclosan with different efficiencies, right? So some of them absorbed it really well, like polyester, and nylon did not absorb the triclosan as effectively. And then we took these fibers that had been exposed to triclosan and incubated them with water from the Chicago River. And we did this for like a month. And what we saw, hopefully you guys can see this better than it looks on my screen. The data for the nylon is to the left, acrylic is in the middle, and polyester is to the right. The dots that are shown in red are no triclosan amendment, and the ones in blue are the ones that were amended with triclosan. And so you can see for nylon on the left, where it didn't absorb much triclosan, we don't see a difference in the communities between the triclosan and the controls. But then for acrylic and polyester that did absorb a significant level of triclosan, we see dramatic shifts in the composition of these microbial communities. So it's a simple experiment that we did. It wasn't very long, but it does show the potential for a pharmaceutical to adsorb to the plastic and then affect the microbes that are colonizing the plastic. And we also can demonstrate some significant changes in the composition of these communities. Just really quick, some things that we saw were significantly lower concentrations of cyanobacteria on the triclosan amended samples and higher concentrations or higher relative abundances of Alphaproteobacteria. So we're still in the process of analyzing these samples. We're also going to be looking at the composition of the algal communities, as well as assessing levels of triclosan resistance and the potential for triclosan biotransformations. So we've shown that there can be an interaction on these surfaces, and then we're going to explore a little bit further what that potentially means for the activity of these communities and their potential for triclosan resistance. We then also, I'll just really quickly mention this mesocosm study that we did in these fiberglass artificial stream systems. In this case, we used a cocktail of 8 pharmaceuticals that are all listed up there, including triclosan again. We amended the streams with either control with no pharmaceuticals, a low level of pharmaceutical concentration, or a 10 times higher concentration, as well as putting in polyester fibers. So we have the microplastics, we have the pharmaceuticals, and they're in these stream mesocosms. And what we saw when we did this, and we added these things independently, we added the pharmaceuticals to the streams, and we added the microplastics and let them just rotate around the artificial streams. We saw that the microplastics did indeed adsorb these pharmaceuticals. And so you can see some of the data we have here for the controls versus the one-time drugs versus the 10-time drugs. So we saw binding of these pharmaceuticals to the microplastics when they were exposed in this artificial stream system. And then we saw that the adsorption of the pharmaceuticals to the microplastics led to decreases in the diversity of the bacterial communities that were attached to the plastic. So these data show the alpha diversity data for my bacterial communities. And so you can see as we go from left to right, we're going from control to the low pharmaceutical concentration to the higher pharmaceutical concentration. So again, the absorption of the pharmaceuticals to the plastic is affecting these microbial communities. It's decreasing their diversity. And again, we saw significant shifts in the composition of the communities. The blue dots represent the high pharmaceutical concentration. The red are the controls, and the green are the lower concentration of pharmaceuticals. So again, the absorption of the pharmaceuticals, is affecting the composition of these communities. And again, we're still in the process of analyzing these data. We're still conducting analysis of the algal communities, as well as doing metagenomic analysis for looking for differences in the potential metabolic pathways of the microbes on these microplastics, and as well as potential linkages between the specific pharmaceuticals that we used and the microbial community responses. So that's what I'm talking about. I'm interested in trying to explore the potential for microplastics to be hotspots for these kinds of interactions. These are the data that we have so far. This is work that we're still following up on. If anybody out there is interested in this besides me, I would really be interested to talk to you or potentially look for ways to collaborate on some of this stuff. I think it's an interesting topic. Maybe somebody else does too. I'd be happy to talk more about it. And then I just want to thank a whole bunch of students and collaborators that I worked with, as well as funding from the Illinois Indiana Sea Grant. Thank you guys for that. NSF and the Illinois Water Resources Center. And with that, I think I've got like a minute or so. I could take questions. Hope that wasn't too fast.
I'm interested in why you chose those plastic types.
So why I chose, because from what I've read, they're pretty common in textiles. So we're cutting them into fibers. So I was trying to model microplastic fibers, trying to model the stuff that would be in clothing that would potentially be ending up in domestic wastewater and having the chance to interact with the pharmaceuticals.
And then your absorption too, that was to virgin plastic, correct?
Correct.
Yes, because we know that the biofilms also.
Right, so that's the other thing, right? If we did it the other way around, if we let the biofilms form first, and then did the absorption, that could be another interesting way to do the experiment. We did it with the plastic alone, allowed the absorption to happen that was in the little microcosm study. We did the adsorption with the triclosan first, and then did the colonization. In the streams, it was all at the same time. We threw everything in at the same time and let it all happen at the same time. But yeah, the biofilm colonization could then also obviously impact the adsorption. So it's a really complicated process. Yes. The 1X was environmentally relevant. We took data from the literature to see what the sort of concentrations would be, as well as some study we've done in the suburbs. The 10X1 was 10 times higher, right? To sort of see like where it could go in the future, and also just to sort of see if we were going to get, I wasn't sure we could get a response at all. I wanted to see what would happen. So we want to sort of model what sort of a worst case scenario thing might be. The other funny thing I'll mention really, really quick, we used City of Chicago tap water to set up the artificial streams that we dechlorinated. There were some pharmaceuticals down in the plastics that we didn't put into the streams. that must have been in the water that got stuck to our plastic that we didn't add. So like, yeah, well, also the thing, well, there's also, we put in sediment from the river, so they could have also been coming from sediment, probably more likely than the tap water. But there was stuff in there that we didn't even add. So yeah, stuff's everywhere. Great, thanks.